Very welcome to this FRAM seminar. FRAM is the Center for Future Chemical Risk Assessment and Management Strategies, and my name is Daniel Slung. I will try to moderate here. So today the theme is micropollutants and the environmental microbiome, an eDNA perspective. And the presenter is Dr. Pedro Inostroza, who is both here at the University of Gothenburg, the Department of Biological and Environmental Sciences, and at RWTH Aachen University. And Pedro has done a lot of eDNA analysis in FRAM-related case studies in Kenya, in Chile, and in Sweden. So we very much look forward to listening to this presentation. So welcome, Pedro, and please go ahead.
Yeah, thanks very much. Thanks, Daniel, for inviting me. And I think, like Daniel mentioned, I'm really happy to have the opportunity to show you some selected results from the FRAM project. I was involved since 2018 in the FRAM project. I'm actually participating in each of the case studies, like Daniel mentioned earlier. So one of them was the Chilean case study, the Swedish one in Stenungsund in the coastal area, and in the Lake Victoria River Basin in Kenya. So the idea of the talk, I think I want to give a little bit of a introduction related to chemical pollution and why we are using that tool of environmental DNA to basically characterize a certain group of organisms that we believe that they have been overlooked a little bit from the assessment perspective and from the some current regulations, particularly in Europe. So far, we have a very good overview about the number of available chemical register in the CAS system. The CAS system is a system where every time that a chemical is developed or discovered is registered in this system, and roughly today we have more than 180 million chemicals being registered in the CAS services. From those chemicals, roughly 340,000 are chemicals that are commercially used. And from those chemicals, about 100,000 are today, or roughly about 100,000 are registered under REACH. REACH is the regulation responsible for registration, evaluation, and authorization and restriction of chemicals in the European Union. However, from, let's say, in the context of the European Union, from these 100 or roughly 100,000 chemicals, there are about 10 to 15% of these chemicals that are environmentally relevant. So those are chemicals that are expected, or we are known that they end up in the environment and actually might cause an effect, an adverse effect. We have a lot of challenges related to those chemicals ending up in the aquatic environment. So the first thing that most of them are at very low concentration. We also are a bit of it blind regarding all the possible transformation products. And one of the big challenges that all these chemicals, they don't occur in a single manner. Rather, they co-occur, making up the concept of this complex mixture of chemicals in the environment. Now, the talk itself is particularly focused in one group of these chemicals, this micropollutant, and it's very important to understand what do we mean with this group of chemicals. So basically, micropollutant is one group of synthetic organic chemicals which actually are present in really low environmental concentrations. So those chemicals are actually in the range of the picogram, nanogram, and sometimes in the microgram range in the aquatic environment. So these are concentrations that we usually measure them, and basically those are a group of chemicals that are present at that range of concentration. Of course, this concept is very instrumental since it's very connected to the analytical capabilities. And of course, probably in the future, we might be also able to talk about nanopollutant or picopollutant, but I think this has been a concept coined already roughly 20 years ago and is widely used today. But you might be wondering actually why to focus in this group of chemicals, right? So I mentioned that roughly 10,000, we expect roughly 10,000 chemicals being relevant in the environment? And the answer is relatively simple and is related to their toxic potential. So these chemicals have the capability to jeopardize the health of the aquatic environment. And this is quite interesting because indeed recent surveys have shown that about 90% of Europeans are quite concerned about the impact of daily use chemicals on the environment, and up to 84 are worried about the impact of this chemical on their health. So clearly there is an increasing awareness about the impact of daily use chemicals in our life and surrounding. Now, who are these daily use chemicals? So these are basically chemicals related to household products, pharmaceutical drugs, and pesticides. So these are basically one of the three main groups, together with also industrial chemicals that we can categorize or make part of this group of micropollutant. Particularly relevant actually are pesticides and pharmaceuticals, since these chemicals have been designed to actively interact with biological receptors, while for household product, they're actually not really, we don't really know exactly their mode of action in some cases. The problem actually occurs when these chemicals end up in the aquatic environment. So they have this capability to distribute themselves according to their physicochemical property between the different environmental compartments. So once they enter the aquatic phase, they might remain in the water compartment. They can also sink and end up in the sediment, while they can also actually migrate to groundwater sources. So that, from a drinking water perspective, may jeopardize also the environmental health of the our population. On once these chemicals actually are in the water phase, some of them have the capability to be absorbed by other organisms, which actually once inside of this organism, they undergo toxicokinetics processes, which include absorption, the distribution of that chemical within the organism, the metabolization of these chemicals, and finally the excretion of them. But they also go through toxicodynamic processes, which actually refers to the specific interaction of the chemical with a molecular target. So once the chemical is inside of the organism, they can interact with enzymes, with proteins, and actually with the DNA itself, causing biogeochemical and physiological changes, actually resulting in a toxic effect. Toxic effect which is unwanted, because unfortunately these organic chemicals, including the micropollutant, have been identified as one of the main drivers responsible for biodiversity decline in the aquatic environment, together with other environmental stressors like habitat fragmentation, climate change, invasive species, and overexploitation, which is quite concerning that together with habitat change or habitat fragmentation, chemical pollution have shown a very rapid increase in heat impacts on the aquatic environment, with profound impacts on particularly on rivers and the coastal area. So in this context, the European Union has established a directive whose aim is to achieve good status for all aquatic lakes and transitional water in the European Union. But what do we understand by good status? So basically, on one hand, we have the aim to achieve a good chemical status. So there's basically concentration of chemical that they don't exist as a specific environmental quality standard, but also a good ecological status. So actually the quality of the structure and functioning of the aquatic ecosystem remain in a good shape, which is very difficult to define. I mean, we have indexes related to that, but it's it's a process itself which demands a very big effort from the biological perspective. Unfortunately, around 29% of the surface water are in a good chemical status in the European Union, and about 37% are in a good or better ecological status. However, it's important to highlight that the ecological status is based on certain selected biological quality elements like phytoplankton, microphytes, phytoventus, benthic invertebrate, and fish. and overlooking certain relevant group of organisms. We also believe that this picture of relatively middle poor chemicals and ecological status quite consistent in other regions, since the chemical profiling today are quite alike between the different places. And a very good example is actually every time that you go to a supermarket in Europe or in South America or in Africa or in the North America, you end up finding the same product in the shelves. So cleaning products are relatively the same, different brands, but the same, roughly same composition, the same with the use of pharmaceuticals. So at the end, all these chemicals end up in the aquatic environment having quite alike chemical fingerprinting. One of these group which has been overlooked from the biological quality element are the microbial communities. Microbial communities are complex consortium of microorganisms, including bacteria, archaea, fungi, protists, and viruses, which actually live together in a specific environmental niche. This group of organisms are quite relevant since they are the base of food webs. They are quite relevant from a biochemical perspective in the nutrient cycle. They're also quite important from a pollution perspective since the exposure to antibiotic has speed up the antibiotic resistance in the environment. And ultimately, sorry, there are still biodiversity gaps regarding their use, their potential use in biotechnology, remediation, and so on. So having this context, FRAM have like a overarching objective to assess the ecological and chemical status of selected aquatic environment around the globe. And although look very ambitious, we can clearly see now here in this image the places in where FRAM actually make a quite tremendous effort in order to conduct field observation together with intensive sampling in different regions. So one of them is South America, particularly Chile. We have also the Kenya, in Africa, and Sweden. And an additional project related to the FORMAS funding, we're also exploring other ecosystems in Europe, for instance, in Germany as well. So we were having the capability to go there, take environmental samples, measure chemicals, and actually generate ecological profiling about the microbial communities in those selected ecosystems. In order to do that, we basically use what we call cartridge. Very simple. So in your right hand, you can see this type of SP cartridge is a type of specific cartridge we use in environmental chemistry, which allowed us to actually concentrate all this micropollutant after a massive filtration, which usually lasts a couple of hours since we want to collect as much water possible and in order to have a comprehensive characterization of the micropollution profiling. And in your right, you can see something quite similar, but actually help us to collect what is called environmental DNA. What is environmental DNA? So environmental DNA is no more than the genetic material released by organisms into their surrounding environment. Um, sorry. This genetic material can be collected using a specific type of filters. So we have in that photo, for instance, one of our master students from the Master of Ecotoxicology at the University of Gothenburg, collecting some water sample for later on actually passing through that volume from the filter that I have shown previously. In the different places that we visit in South America, in Africa, and also in Europe, we use a very similar sampling setting. So we did a biotic characterization of the water samples that we collect there, but also we did a biotic characterization. So on one hand, we collect between 50 to 100 liters of water for micropollution profiling, which later on were actually measured using a technique called target analysis for roughly about 750 chemicals that we were interested to screen. And beside that, we also did the very traditional environmental sampling where we took water samples well for nutrients and metals and temperature and the typical physical chemical parameters. Regarding the biotic characterization, depending of the country that we visit, we did in some of them freshwater and marine sampling, on some of them both, and also we took different type of environmental samples. So we took water samples for characterizing microbes in suspension. We also collect biofilm, like you can see in the photo here. So basically, it's a group of it's a structure which allow the microorganisms to remain there and to create a sort of complex colony in this very defined structure called biofilm. But we also uh, collect sediment samples as well. So we try to have a comprehensive sampling also of the environment, having different compartments. Later on, we also use very specific biomarkers in order to characterize bacteria, fungi, and protists. When we talk about micropollution, so basically who they are. So you can see here, hopefully in the right side, a very long list of different micropollution classes. So you can see in blue pharmaceuticals, in green you can see the pesticides, and then personal care products. So we are always allowed, or we have the capability actually to characterize them. And this is a good example, for instance, how the River Aconcagua basin in central Chile look like. So we have three pie charts basically where we were able to characterize area with low anthropogenic pressure, agriculture pressure, and agriculture combined with urban pressure. What is quite interesting, or actually now, like I mentioned, we started in 2018, so now we can have a very big and comprehensive picture of the different sampling sites. Is a quite consistent pattern in where we observe a very strong composition of basically micro pollution being characterized by pharmaceuticals and pesticides. So most of the time, those two groups made up to roughly 50% of the micropollutant that we were measuring in the different places. So like I wrote here down that the chemical profiling is quite similar also in Skåne area in the south of Sweden, in one particular river in Germany, where we were collecting samples there, also in the northern Patagonia and in Kenya. So the chemical profiling is very, very similar, being the pharmaceutical and pesticide the two top classes of chemicals. Like I mentioned also earlier, we also took samples in rivers and in the marine environment, in Sweden and in Chile, in the northern part of the Patagonia, and we also observe a very similar chemical profiling. So again, pharmaceuticals and pesticides are quite dominant group of chemicals in the coastal marine environment, which is quite interesting because the profiling of micropollutant in coastal area is very restricted. So most of the information that we have is mainly from surface running water from North America and Europe and Asia. And actually studies characterizing coastal pollution from a micropollution perspective are quite limited. So we were one of the first group actually doing that for instance, in rare environments like Patagonia or even in some fjord system in Sweden. Just to give you a little bit of context, what we can do with that type of data. So this is an ongoing study in where we are taking all the measurements from Chile, from Kenya, and from Germany, Sweden, and also in combination with other studies in the European Union. And if you focus in the last three bar plots, both for algae, crustacean, and fish, we are actually able to do risk predictions. So this is an ongoing project in collaboration with colleagues from UFZ in Germany, where actually now we're making use of the data that we've been collecting through FRAM, but also other projects in order to have a more continental or more, let's say, continental assessment of micropollutant in surface water. But like I mentioned earlier, I would like to focus in some of the case studies that we have in France. So one of them is the Stenungsund case study in Sweden, in the western coast of Sweden. What we did that there, actually, we went there and we had the possibility to take both marine and freshwater samples. So hopefully you can see in the figure here, all our marine sites are labeled with M. So we have from the marine site M1 to the marine site M6, running across the fjord, actually trying to characterize the likely impact of the Stenungsund industrial hub. So Stenungsund is one of the areas in Sweden which hosts one of the largest petrochemical industry in Scandinavia, and we were quite interested to characterize chemically the fjord, but also ecologically in order to assess if the chemicals likely entering the fjord may have an impact on the aquatic microbial communities. But we also had access, we were quite fortunate to have access to one of the wastewater treatment plant in the Stenungsund area. So we also took sample actually from the marine outflow. So basically uh, fresh water that end up in the middle of the fjord. This is F3. So actually the marine outflow is very close to the sampling site M5. So we have this sampling setting in where we have some freshwater input, all the F sites, and all the receiving, very close receiving marine site in M. And then basically we took water sample for chemical profiling and also for microbial profiling. And what did we observe there from an environmental DNA perspective? So these are figures related to biodiversity. So this is one of the metrics that we usually use in biodiversity. It's called observed biodiversity. I just showed these two here for two different groups, the bacteria and fungi, but the pattern is quite consistent for other metrics. Usually we do a multi-metric assessment, and just for the sake of the presentation, I'm just showing the observed, but the pattern for Shannon or Cow1 is basically the same. So what do we observe there? So you can clearly see here now all the marine sites are clustered together here, and they have actually a lower biodiversity to the freshwater side for bacteria and for fungi. But which is quite interesting to observe is the biodiversity of bacteria is very, very low in comparison to the two natural streams, so which make a lot of sense. So we would expect that the biodiversity coming from a wastewater treatment plant to be low, uh, but still is quite high for bacteria. So you can see now in the right figure how the biodiversity for the group of fungi is very low in comparison to the other sampling site. The other finding that was quite interesting to observe is this sampling site here, that is the M5, that is the sampling site really close to the marine outflow. So you can clearly see how that sampling site, so basically very, very close to the freshwater treatment plant outflow, have a massive increase in biodiversity in comparison to the other marine sites. But we don't observe such a similar picture for the fungi. So still you can clearly see how the sampling site very close to the wastewater treatment plant is slightly showing a higher biodiversity, but not as the bacteria group. This is quite interesting because, like I mentioned before, there are not so many studies actually characterizing microbial communities in a fjord system, and beside that, on top, with a clear input of micropollutant into the fjord due to the presence of a wastewater treatment plant there. Sorry. We were also able to characterize these two communities, the bacteria and the fungi, from a compositional perspective. So just to make the explanation simple, I have placed this orange arrow here so you can clearly see how the sampling site F3, which is the wastewater treatment plant effluent, show a composition quite different to the other sampling site, the other freshwater sampling site F1 and F2, and the sampling site FM5, which is again the closest one to the wastewater treatment plant effluent, overall have a very similar composition to the other marine site, although we can observe some increase in abundance of very selected group. The same picture, or quite similar picture, we observe as well for the fungi community. So you can clearly see how certain group have seen their abundance or the relative abundance shrinked. Like for instance, the Basidomycota group here in blue, while other group with really low abundance in gray have seen their abundance quite increase in the wastewater treatment plant effluent. And similar idea with the bacteria. So generally very similar pattern between the marine site, while really specific changes here in the top in red, particularly in some group for the fungi. When we wanted to know how similar or dissimilar the places were, so it's very important for us to know if we have a very homogeneous environment or some places actually become very different because of the environmental stress. We conduct this type of analysis in where it's called beta diversity analysis, in where we can clearly observe the differences between the different sampling sites. So here you can also have highlighted with the black arrow how the sampling site related to the wastewater treatment effluent is quite dissimilar compared to the other freshwater site, but also quite dissimilar to the marine site. And again, very similar to the pattern of the community composition, how the marine site M5 here is becoming quite dissimilar to the other marine site. And very similar pattern as well observed for the fungi group, while you can clearly see here how the marine site M5 actually is becoming quite similar to the wastewater treatment plant effluent. So until that point, and basically we did that exercise again for Chile, for Kenya, we observed very similar patterns. So basically microbial communities shown a very site-specific changes, and with most of the time, changes in their alpha diversity, in their observed richness, while becoming most of the time quite dissimilar in polluted sites. But we also wanted to try to get a second assessment of how chemical pollution might be driving those changes. And we came out with this suggestion of normalizing environmental concentration of antimicrobials by their toxic pressure. So this is what we call the toxic unit MIC. So basically the toxic unit normalized by the minimum inhibitory concentration of the chemical on the microbial community. That was recently published in environmental pollution. And in the right side, you can see an ordination figure in where if you focus here in the lower right part, you can clearly see this TUMIC. So this is equal to antimicrobial stress. How the antimicrobial stress actually is correlated very strongly with some specific sampling site. And those sampling sites actually are sampling sites that later on we characterize with, for instance, bacteria known to promote resistance, antibiotic resistance, but also bacteria that's a decrease in the abundance of bacteria actually having certain relevant nutrient function in the aquatic environment. For Stenungsund, basically, we observe a quite similar pattern. So you can clearly see here how the toxic pressure, particularly for antibiotic, correlate very strong with the wastewater treatment plant effluent. Basically, that means that it's very likely that antibiotics being released by the wastewater treatment plant is actually the responsible of the structuring of the microbial community in that particular place. And something similar we observe for the fungi. So in the case, we observe a negative correlation between the pressure from the antibiotics and the fungicide in regard to the freshwater site M3. So those kind of metrics help us to actually have some first approach to how micropollutant might be changing the structure of the microbial community, particularly bacteria and fungi. And again, we observe this pattern very consistently in both in Chile and also in Kenya. It's important to highlight certain limitation of our approach. So the first one that yes, micropollutants are only part or they're a fraction of the whole organic pollution universe. There's so many other chemicals there that actually is a very complicated to assess in the context of their impact on microbial community because we don't have ecotoxicological data for that group of organisms, which is actually something very similar for other group of organisms. So we are always also lacking ecotoxicological data for other species like algae or crustacean or even fishes for certain group of chemicals. Another limitation of our approach, particularly, is that we use this target analysis approach, which is like the name said. So we have a predefined number of chemicals that we want to look at. And like I mentioned, so early in the introduction, so we are aware that the aquatic environment, there are thousands of these chemicals, couple of thousand of them. So here we're basically really picking a few of them, although our target list was quite large with roughly 800 chemicals, still quite a lot, but still the approach itself is really limited to building up those lists. Certain other limitation is related to the environmental DNA approach. The environmental DNA approach is not able to distinguish whether the DNA that we are collecting is coming from a live organism or from dead organism. So this is one of the limitation of the use of environmental DNA. But today there is a second approach called environmental RNA, which actually have the capability to capture the active fraction of live organisms. So that can be another type of technique that will allow us to actually compare our result with environmental DNA and try to tune better the relationship between the whole DNA and only the active fraction of RNA. Another limitation, actually, but this is not just related to our study again, is that when we do this taxonomic, the biodiversity identification of the organism in our sample, those are always really related up to the status of the taxonomic databases. And we have a massive gap regarding to knowing which are the organisms out there, but this is not restricted to microbes. So we see the same pattern for other microinvertebrates, for fish, even for microalgae. Another limitation is that environmental DNA, but also environmental RNA, they don't account for abundancy data. So that is a bit complicated when we want to contextualize our results towards more regulatory perspective, because of course, current legislation requests to also know the abundancy of those organisms in order to develop indexes, so ecological indexes. And last but not least, is that the environmental DNA is restricted to just structural assessment. So we are also overlooking the functionality of the community itself. However, I think although I have mentioned many limitations, which actually not just related to our study, I think it's limitation from about the field. We have very important take-home messages. For instance, that environmental DNA is able to actually provide strong evidences related to changes in the community, microbial community due to the chemical pollution pressure. Those changes seems to be really site-specific, which actually could be very beneficial because they seem they're not widely expressed in the ecosystem. Probably certain mitigation strategies or management can be developed around those places. Now, metrics or like a community composition seems to be quite good, actually informing about those changes. and as well as the use of metric related to the degree of the dissimilarity. Another take-home message is actually we were able to show, not just in Sweden, but also in Kenya and in South America, Chile, a strong correlation between those dissimilarity changes in the microbial community due to micropollution. With that, I would like to thanks to all not to the all collaborators involved in all the different case studies. So this is quite short. So there's a lot of people in the back, and particularly the master student from the ecotoxicology master program at the University of Gothenburg, because they were really supportive, and many of them actually have done their master thesis in the context of the FRAM Center and the case of the different case studies. So yeah, so you have any question? Yeah, I think we can move in that direction.
Yes, thank you so much, Pedro, for this very interesting and informative presentation. I think we can now close the recording for online viewers. We say thank you, and I hope to see you again at our next seminar, which will be on, is it June 5th? I think, where we will talk about regulating cumulative impacts on coastal waters. More information will come on this. So thank you very much.